My heart was trying to take care of patients with epilepsy who unfortunately don't respond to medications, and beyond medications options, we have the stimulation. And that just bring me here almost 10 years later when I start looking into that, how many questions I had, how many questions I could not find anywhere. So I thought it was an excellent path.
This is Bold Minds, Future Leaders in Canadian Brain Research. I'm your host, Fiona Sanderson. I work at Brain Canada, where our mission is to bring together funders and researchers to enable health innovations for Canadians. The Future Leaders program is made possible thanks to an anchor gift from the Azrieli Foundation and matched by Brain Canada through the Canada Brain Research Fund. Come along with me as we journey into the bold minds and labs of researchers who are redefining our understanding of the brain. Your brain is an incredibly complex network. The firing circuits of your brain and nervous system shape everything from what you'll eat for breakfast to keeping your heart beating while you sleep. Today, we're zooming in on what happens when those circuits are activated and when they go awry. And how are researchers learning to fix them? Whether it's using vagus nerve stimulation to safeguard people with epilepsy from the worst outcomes, or exploring the hidden machinery behind confidence, decision-making, and how dopamine shapes the way we learn. There's still a lot we don't know about these networks. I'm joined by two researchers at the cutting edge of brain science, in two very different arenas, but sharing one powerful idea. If we better understand the circuit, maybe we can change the outcome. So let's get into it. This is Bold Minds. Today I'm joined by
Anna Suyer-Marti. I'm an epileptologist and associate professor at Western University in London, Ontario.
Her team uses targeted brain stimulation therapy along with brain scans and electrical recordings to treat people with epilepsy, improve their health outcomes, and understand the neurobiology behind these processes. I'm also joined by
Paul Masset. I'm an assistant professor at McGill University in the Department of Psychology and an associate academic member at Mila Quebec AI Institute.
His team is interested in understanding what happens in our brains when we sense our environment, make decisions, gain confidence, and process rewards or deterrence. They use rodent models, complex behavioral tasks, and brain recordings, together with AI, to understand how differences in the brain's circuitry contribute to cognition and health and disease. Anna, Paul, welcome to Bold Minds.
Thank you.
Thank you.
So Anna, let's get started with you. You study epilepsy, and about a third of people who have epilepsy have a drug-resistant form, meaning their seizures are not effectively treated with a currently used drug. So can you walk us through exactly what's happening in the brain when someone's having a seizure? What are the long-term implications or risks for someone with untreated epilepsy?
So epilepsy can be simplified as an abnormal electrical activity in the brain, and this interferes with cognitions in mood, autonomic functions during the seizures, of course, and also in between the seizures. Furthermore, there is the possibility that they suffer what we call sudden unexpected death. In epilepsy is a fatal complication called SUDEP. So we need to be extremely careful and diligent to manage our patients. That's the reason we offer different types of strategies, including neurostimulation as one of my main areas of research. It's a technique that applies electrical stimulation to some different parts of the body, mainly in the nervous system. It could be in the vagus nerve or inside the brain. So we are using some of those techniques to try to modulate the seizure activity, to try to reduce this abnormal electrical activity that is happening. Epilepsy is a disease that it presents as recurrent seizures, but it's associated with changes in neurobiology, psychological, social, and cognitive. So what it means is in patients with epilepsy can struggle with other things, like maybe memory deficits, with mood disorders like depression, anxiety, or even sleep problems or feeling isolated. So all these, what we call comorbidities, directly impact to the life of our patients with epilepsy. At some point, some of the patients even refer that it's even worse having severe depression than having even recurrent seizures. So that's something that most of the efforts of the epilepsy community have been focused in seizure control and has been kind of ignoring all these other conditions going on in our patients. In the most recent years, when the epilepsy group start to think, oh, we should start paying attention to the other things and how we can help. So not easy task, but we are trying our best to improve lives.
So you mentioned that dysregulation of the autonomic nervous system leads to an increased risk of sudden death in epilepsy, or SUDEP. So can you walk us through this? Like, what is the autonomic nervous system? What does it mean when it's dysregulated? What can be done about that for patients?
So this is a brand new area of research that we start exploring through collaborations with other scientists. And of course, as we know that the brain is connected and regulates different organs or all the organs of the body, and how these organs may respond differently to our brain. So the implications are still, it's a chapter that I'm trying to understand better, but perhaps that could affect the response to, for example, the heart response or the breathing response. And of course, after a seizure, many of these organs get affected. So the idea is to understand if normalizing the autonomic nervous system, we will be reducing the risk of dying after one of these seizures. So it's not an easy chapter, but I hope that through some of this research, we can answer more of your questions.
So, Paul, you're also looking at a sort of cascading of neural signals through circuits, but this time from one part of the brain to another, and for a very different purpose that has to do with dopamine, learning, and confidence in our decision-making. Tell us a bit about your research.
So my research is sort of quite diverse. It sort of bridges the computational neuroscience part, where we're sort of thinking about what are the computations that brain circuits are doing, and the experimental, systems neuroscience, which is let's have, in our case, of an animal model, do a task, see what the neurons are doing while the behaviors are occurring. And it's trying to bridge that gap to what is the circuit, what are the neurons doing while there's a behavior, to thinking about what are the computations and what are they doing while this behavior happens. And one of the key sort of directions in the lab is thinking about what is the role of dopamine in learning, as you mentioned. And dopamine is very important because a large amount of psychopathologies and also some neurodegenerative diseases, right, like Parkinson's disease, are basically linked to dysregulation of dopamine signaling. Basically, dopamine signaling is not occurring as it should, and that leads to disease. But it leads to very different sort of issues. And so one of the key questions in the lab is what is sort of the function of the heterogeneity, right? You have this neurotransmitter, dopamine, and we're sort of really interested in how it acts in a part of the brain called the striatum, where dopamine is sort of released there, and it helps modulate the synapses and basically learn associations between events. But even though the striatum is sort of one structure, it is not homogeneous. It has different inputs. Different parts of the striatum gets input from different parts of the brain and different parts of cortex. And the question is, how does dysregulation in one part versus the other leads to different symptoms, right? Why is disrupting this specific part of the pathway leading to this specific type of sort of symptoms and disease? And that will help us understand basically what goes wrong in that disease. And so it can come from different origins, right? For example, in Parkinson's disease, it's really the neurons that sort of produce the dopamine and release the dopamine in striatum. These neurons themselves die. So obviously there's less dopamine around. And for example, one of the sort of therapies is to sort of give dopamine precursor. And so you increase all the levels of dopamine. And so you improve on the symptoms, but you also have side effects because you didn't just change dopamine back to sort of vaguely normal levels in the pathway you're interested. You also increased it in other areas where it was not disrupted. And so now it is disrupted in some ways. And so that leads to other side effects, right? And so understanding basically how the same circuit, the same architecture, same types of neuron do completely different things depending on, for example, the inputs they're getting, right? So one of the ideas, for example, is that you're getting inputs and you're learning about something that is motor, right? And so you can think in Parkinson's, you're learning about something that is motor actions. You can have the same architecture, the same way of learning, but now if your inputs are not motor, they're, for example, sensory, then you could have another problem and you have issues about learning about sensory things. And it's thought there's starting to be evidence that part of the striatum that is more linked to sensory areas might be involved in, for example, hallucination-like behaviors in schizophrenia, right? And so you have the same dysregulation at the local level, but because the circuit is in a different place in the brain, it just leads to completely different outcomes. And so understanding the fundamental computation might be absolutely key to understand how to treat these super different sort of pathologies as not one, but at least having some common principles.
That's interesting. So there are a lot of different characteristics or personality traits that are commonly ascribed to people. And I think many can be boiled down to our decision making. So things like people who are risk takers versus risk averse, or those that crave a short-term reward versus those that like a slow burn or a long-term satisfaction. So are these things all related to the way our brains process dopamine? Are they more or less hardwired into our brains?
So whether they're hardwired, that's a very difficult question to answer. Another type of diversity that I uncovered in my postdoctoral work is like looking at these dopamine neurons. We identified that different dopamine neurons actually do this learning, and they basically compute the error that is needed for learning, right? The discrepancy between what you expect and what you get. And that's what the dopamine neuron computes. We showed that this error signal is computed at different timescale because basically the neurons, they compute the value of things or the signal they're computing about over some time horizons, like how good are things in the near future, right? But what is near? Near has very different definitions. It can be the next second, the next 10 seconds or something like that, right? And so what we identified is that different neurons have different timescales. And so from that, you have this very tempting hypothesis to say that, oh, okay, if you have more short timescale neurons, you're going to be sort of impulsive because you're going to only learn about short timescales things. If you have long timescale neurons, it's going to be more long-term. So this is like a very tempting hypothesis, but it's sort of a working hypothesis at the moment. But we do see that the neurons are doing this. So there's starting to be this mechanism that potentially could implement this sort of things. And there is ideas that some of these things, like, what you're talking about, time interval, the idea of temporal discounting, how much do you devalue things in the future, does change in addictive behavior, for example. And so you take sort of cohorts of patients that have a specific addiction versus not, they tend to potentially have a sharper discounting. They're sort of more myopic in the temporal horizon. They think about more near-term things. The question is a bit the chicken and egg, right? Are they addicted because they have this thing and that they cannot sort of plan ahead and see that it's not good to do something because it's going to lead to sort of issues in the future? Or is it the addiction that created that? They're still very much open questions, but there's starting to be ways to measure this and potentially quantify this in a way that is quite sort of, I want to say, computational, and we have sort of a grounded theory of why would you even do this sort of behavior.
I'm curious, when someone has a seizure or has a series of seizures over their lifetime, so this flurry of electrical activity that's happening in the brain, does that change the neurotransmitter milieu? Like when you have all of these synaptic connections being activated, what happens there?
To learn that, it's a bit tricky because we would like to see over time. And one of the ways that we can assess some of those changes using MRIs, and we can see that over time, some areas may shrink or becomes more atrophic than other areas. And of course, having less tissue may be associated with less function in some of those areas. It doesn't mean that patients who suffer seizures through the entire life will have an atrophic brain. But in some cases, we have seen in some specific conditions like mesial temporal sclerosis, other specific types, that you can see a change. And of course, that, as I say, impacts function, and the function is likely related with some neurotransmitters change. So, but again, we cannot generalize, as epilepsy is very heterogeneous disease. And from presentation, like as you say, you may have 2 seizures in your lifetime, or you may have tons of seizures through your life. So that's very different scenarios, and as well as the response to medication and other comorbidities, or the reason why you have epilepsy could be different, and that could accelerate some of those processes, or maybe nothing changed. That's one of the biggest challenges, this complexity in the disease, presentations, evolution. So that's the reason we always are looking for more answer and more options for our patients.
I think one thing you pointed out is, and I think is very important in neuroscience, is the idea of heterogeneity, right? It's like these things we classify them as the same, but they're very, very different, right? Like if you think in sort of more molecular biology, right, like cancers, you have a cancer of something, they're actually very different. And we understand a bit better there because we can sequence how different they are, right? But in neuroscience, we're still quite far from that. We're getting there, but we're still far, and we still classify as one thing, things that are intrinsically different. They just manifest themselves in potentially the same way, but they're like intrinsically very, very different.
Yeah, I completely agree that in epilepsy, at least, is having one single model doesn't work. You can have one single model with one type of epilepsy, and despite that, 2 patients with the same similar presentations will have very different evolution, different response to medication. So again, it's a bit challenging, this field.
So you both study neural circuitry that has distal effects, so meaning something happening in one spot that has an effect some distance away, be it all the way from the gut or the heart to the brain, or from one part of the brain to another. Does understanding these far-reaching effects, both literally and metaphorically, hold promise for the future of non-invasive modulation therapeutics? So, that could be to control a brain disorder, or even just to boost how we learn and form memories?
Again, the dimension of these therapies is still unknown. And part of my research is, of course, in this project was to assess the autonomic impact of the vagus nerve stimulation. But in other areas of research that I'm doing is looking into other comorbidities very frequent in people living with epilepsy. So it could be from mental health to sleep disorders. And of course, this goes back to the initial questions about how the autonomic nervous system maybe modulate all of these functions. And it's something that we cannot try to put in compartments. We focus in mental health, we focus in sleep, but perhaps there is an interrelation with all these functions and how this could be modulated. So how at least I see neuromodulation has been proposed, for example, vagus nerve stimulation in clinical settings for different conditions, like from severe depression, from obesity. It's hard to puzzle how this same function works in so many different diseases, but perhaps there is some root that it's all indirectly responding to the same way. So I think we still don't have the right answer. We hope to have it in the future, but the interconnections and the distal effect, it's obvious that we are seeing.
Yeah. So I think the key thing there is the invasive versus non-invasive, right? So I think specifically if you think about dopamine signaling, for Parkinson's disease, you have sort of deep brain stimulation. So it's invasive, but you are modulating basically those nuclei and then it like making the effects on the learning. You're sort of boosting the activity of the neurons. And it does work really well for Parkinson's disease and for other diseases as well, right? But this is very invasive techniques. They're sort of last resort things. One of the things with the dopamine system, right, like the dopamine nuclei are in the ventral tegmental area, the substantia nigra, they're very deep in the brain. So it's sort of hard to reach them without like going inside. The striatum is also a bit deep. So all of that part of the circuitry is hard to access. That's where you would need really to understand how is the learning happening, because now you're not changing the learning itself. You would be changing sort of what are you learning about.
It's so interesting that, all of these things are controlled very physically in our brains. So I want to talk a bit about how you got where you are. Paul, how did you end up at the intersection of neuroscience, AI, and psychology?
Yeah, so that's a good question. So I started sort of my undergrad as an engineer, thinking I was going to go in some sort of, heavy engineering things like, I don't know, like designing airplanes or something like this. And then I got brought into the more computer science side of things. I got interested in that. And then I had some very interesting lectures sort of people who were exactly at that interface, who were in the engineering department, but who were neuroscientists. And so who basically were talking about bringing ideas of sort of Bayesian inference. How do we compute probabilities correctly? And how can that basically help us understand motor control, visual perception, and all of that? And so that sort of got me into that part. And then I continued, I did a master's in sort of computational neuroscience to learn more about the neuroscience as well, right? Because I had done some classes, but not that many. And that really got me into this. But then I've had the feeling that I couldn't stay on the computational side only. I had to actually understand what was happening. And so for my PhD, I looked for a place where I would be able to sort of be in an experimental world a bit more and do experiments. And so that's where I did some sort of, rat behavior, decision-making, recording neurons in prefrontal cortex, looking at this idea of representations of confidence while the animals are behaving. Also, AI has changed tremendously in the past 10, 15 years. And so there's all these ideas about, it's not necessarily that the brain is not doing what the algorithms are doing, but the algorithms show us how information can be transformed from inputs to pretty complex outcomes. And so there's at least ideas there that we should look at to try to understand how the brain can do it.
So it's not just that we're using the brain as a model for what AI should do, but now we're doing the opposite. We're looking at how AI does things to learn about how the brain might be doing them.
Exactly. So the idea is to also go the other way, to think about how the brain is built and how can that help us do better algorithms. But in a way, that has worked at a very conceptual level. It's not like you go to school for 20 years and then you stop learning. That's something that actually algorithms are not great at doing at the moment. You sort of train them, put them out. And so there's clearly inspirations that should be looked at. But what is the right level of abstraction, I think, is where the big question is at the moment.
Anna, your research background was in headaches and migraines. So what drew you to epilepsy and neuromodulation therapeutics?
I became interested in epilepsy very early on in my residency. So during residency, I already started doing, of course, passion about the patients, but also starting to do some small projects in epilepsy and presenting in different national and international meetings. But unfortunately, when you finish residency, it's not what you want. Sometimes it's the options that you have. So a close field that I already enjoyed was also headache. So during my master's, I focused in headache. And the lucky part is with my mentor in headache, he was an expert in neuromodulation. So he was applying neuromodulation or stimulation to manage headaches. And we published a few papers about that, and I managed a few of those devices. But unfortunately, my heart was trying to take care of patients with epilepsy. So that's when I decided to make the big jump and move to Canada and pursue a fellowship in epilepsy. And then I get to learn about these patients who unfortunately don't respond to medications, and beyond medications options, we have the stimulation. And that just bring me here almost 10 years later, how much we don't know. When I start looking into that, is how many questions I had, how many questions I could not find anywhere. So I thought it was an excellent path, learning more about these devices that commonly we are using.
I think this is like a quintessential research experience. There's no shortage of questions. You just have to find the path that sort of speaks to you, right? What you find to be the most interesting is where you're going to have the biggest passion and answer those questions. So I love hearing that. You're both doing really cutting edge research with pretty novel techniques. Is there something you believed about the brain early on or when you started that you've since changed your mind about with the advent of new tech or new information?
So I think one thing in the field I am, right, where we sort of look at animal models and we record the neurons, activity of single neurons. One of the sort of transformational technologies in the past, like 5-10 years, when I started, you basically were recording from like a few neurons at a time, right? You had an animal, you record like 4 neurons at the same time. You're super happy, right? It got better. You had some slightly better methods. You could get 20 neurons maybe at the same time. And so now you're starting to get several single neurons and see how they behave together. People had some ideas about what could do, but they were looked at it as independent entities to some extent. And now basically there's imaging techniques, recording techniques. You can record like thousands, tens of thousands of neurons, single neurons, right, at the same time from different brain areas while an animal is doing a complicated task where they see something, they have to decide what it is, and then make a choice. And so now you have this population level view of things, right? I always felt, because I came from the more computational side and that before I got into the experimental, that the single neuron view was obviously not the right thing completely. Like, it's very important because in the end, single neurons are the ones implementing the computation. They are sort of protein machines. And when the gene goes wrong, when something goes wrong, it goes wrong in the single neuron to some extent. But what is happening at the level of the circuit, right? That was hard to know what it was going to be. And I think that's the big change where now we can record this and how are these neurons talking to each other.
Some of the things that Paul mentioned, I can make also a correlation in humans that, of course, trying to record from brain activity, it was nothing new. Like from 1950s, they were already recording brain activity to try to determine where the seizures are originated. But over time, we have been improving the technique, being able to record for longer days, and also better understanding of these recordings. So I think it's quite revolutionary that despite it's not a new technique, for example, for epilepsy, has been modernized, and the ways that we are looking into the data is completely different. Similarly, the epilepsy surgery is not a new technique, but we have been improving significantly how we do it, or we use the new techniques as stimulation. That this has been more in the last 2 decades, how that field has grown dramatically, and how this can improve lives of patients, but also how this may help not just seizure reductions, other comorbidities that we see in our patients.
Really better tools to understand what's going on so that you can ask the questions and, get that information that you need to feed into the research. That's really important. If you could explain one insight from your work that you wish everyone understood, what would it be?
I think one thing is that learning is about reducing the error between what you expect and what you get, right? The learning signal and sort of what you're getting has to match what you expect to some extent, right? And so that's why it's a very adaptive process, and something that works for someone might not work for someone else. I think about it in a very sort of narrow sense, right? That when you have a learning in a sort of neural network or in the brain, right, you're getting inputs from the world, like you're getting something, and you are expecting, you have an internal model of what's happening. So these two have to be different if you want learning, but they can't be too different, right? Otherwise, there's no learning. And so I think this idea of learning as a sort of a prediction error is very important, and thinking about it that way, and that is incremental steps that allow you to build much more complex sort of understanding of the world.
Epilepsy is more than seizures, that there are many comorbidities associated with epilepsy. And when we try to treat, again, 1 of every 3 will not respond to normal treatments. We need to offer other more invasive techniques that, of course, for general population seems a bit dramatic to implant devices or do surgeries. But this has a drastic impact in their lives, from just not the quality of life only, but also the risk of dying. How this works, again, this is kind of a black box still, that we know the input, we know we are looking for the output, but still how the brain is responding to that is still unknown. So again, this is still work in progress. This is unfortunately the reality that we are living. And some of these more invasive techniques are used because there is no other option right now.
I know you both have almost brand new little ones at home. So thank you so much for sharing some pretty valuable time with us. And, I also have kids, not so little anymore. But parent to parent, what do you hope your research can unlock for the next generation?
At least from the epilepsy field, there is a lot of questions and a lot of answers that need to be made. So the idea is to improve life of the people living with epilepsy, reducing comorbidities and reducing the risk of dying from epilepsy. That will be a good goal. But of course, this, as I say, through the complex disease is not an easy task.
I think one thing is like my work specifically is a bit more, theoretical and sort of experimental. So we're not that close to the clinic at the moment. But I think the dream is to see some of these ideas really making an impact in terms of how we think about disease, potentially designing new diagnostics or new therapeutics. We can see path to that, help design a new drug, help design a new therapy, behavioral therapy. And so like seeing some of these ideas come through to actually change people's lives, that would be an amazing thing. And that's the hope, right?
So thank you so much, Anna, Paul, and to all of our listeners today. Thank you all for joining us on Bold Minds.
Thank you so much. It was a great discussion.
It was a pleasure to be here. Thank you for also helping us to learn about other research going on in Canada.
Bold Minds is a Brain Canada production with support from the Azrieli Foundation. Our executive producers are Gillian Donnelly and Kate Shingler. Our lead producer is Jess Schmidt, with editing by Meighen.
Thanks for listening. If you enjoyed this episode, we'd appreciate it if you could send it to a friend. If you want to learn more about Brain Canada and our Future Leaders program, please visit our website at braincanada.ca.